Ever pondered how genetic material is transported from one cell to another? The answer lies in vectors. Now, what exactly are vectors? Imagine a vehicle, but instead of transporting people or goods, it carries a sequence of DNA or other genetic material from one cell to another. This vehicle or vector is often a piece of DNA itself, and its primary role is in molecular cloning procedures where it introduces a desired DNA insert into a host cell. The DNA insert that is transmitted by a vector is termed recombinant DNA, and the whole process? It's known as recombinant DNA technology. So, in the world of genetics, vectors are like taxis, carrying passengers, in this case, DNA sequences, to their destinations. All these cellular operations and processes, the multiplication, expression, or isolation, are driven by these vectors. Vectors, in essence, are the vehicles that drive the genetic world. What makes a vector suitable for its job? Let's delve into the features of vectors. One key characteristic of a vector is its size. An ideal vector should be small enough to be incorporated into the host genome. This compact size is crucial as it allows the vector to navigate through the complex genetic landscape inside a cell. In addition to being small, a vector must also have the ability to incorporate large-size inserts. This means that despite its small size, a vector should be able to carry a significant amount of genetic material. This feature allows vectors to transfer a diverse range of genetic sequences from one cell to another. Lastly, vectors must be able to integrate into the host genome. This integration allows the transferred genetic material to become a part of the host cell's own genetic blueprint. The features of vectors equip them to carry out their role efficiently in the world of genetics. Vectors come in different types, each with a specific role. Let's look at these types. Cloning vectors, for instance, are predominantly used for transferring foreign DNA into host cells. Plasmid vectors, which are small, circular DNA molecules, are widely used in laboratory research for gene cloning. Cosmids, on the other hand, are plasmid-like vectors that carry a COS site, allowing them to be packaged into phage particles for infection. Bacteriophage vectors are viruses that have been modified to use as vectors to clone DNA. Bacterial artificial chromosomes and yeast artificial chromosomes are used to clone very large pieces of DNA. Human artificial chromosomes have potential therapeutic applications. Viral vectors are tools commonly used by molecular biologists to deliver genetic material into cells. Expression vectors, shuttle vectors, and secretion vectors each have their unique roles in genetic research. Examples of these vectors include PBR322, a widely used plasmid vector, PUC19, a commonly used cloning vector, and lambdaphage, a bacteriophage vector. Each vector type has its unique role, making them indispensable in genetic research. The real-world applications of vectors are fascinating. Let's explore some of them. Vectors are instrumental in generating engineered organisms for specific functions. An exemplary application of this is the engineering of E. coli bacteria for insulin production. This revolutionizes the field of medicine, particularly in treating conditions like diabetes. Vectors also play a critical role in isolating a particular gene sequence within a genome. This isolation process is crucial in various research studies as it allows scientists to focus on one gene at a time, understanding its function and potential. Furthermore, vectors are essential tools in DNA sequencing. By transporting particular DNA sequences into new cells, vectors facilitate the determination of the nucleotide sequences, providing invaluable insights into the genetic blueprint of organisms. In a nutshell, vectors have a myriad of applications, revolutionizing fields from medicine to bioengineering. As with any technology, vectors come with their own set of limitations. Let's understand these. One key limitation of vectors is their instability. This can be due to changes in metabolic energy, shifting pH levels, and varying temperatures in different hosts. The stability of vectors also largely hinges on the specific type of vector used and the genotype of the host. Another constraint is the overexpression of certain genes in the host cell, a common issue associated with vector usage. This overexpression can lead to an imbalance in the host's cellular function, potentially disrupting the desired outcome. Additionally, the use of a single type of vector might not be sufficient for a particular purpose, requiring the use of multiple vectors. However, this adds complexity and can lead to difficulties during the process. Despite these limitations, the potential of vectors in genetic research and applications remains immense.